Well, welcome back to a video in which we're going to cover one of the most amazing groups of insects, and I say that as a hymenopterist. Today we're going to get into the Coleoptera, and we'll bring along its sister group, the Strepsiptera, just to keep it company. So let's start with the Streps. I've discussed some aspects of their biology and why they're interesting creatures in the context of the video dealing with long branch attraction, and how this is one of those classic groups where it's just so different that people didn't know, or they had a hard time coming to a consensus on where it really belonged in the classification. In terms of their biology, the Strepsiptera are another parasitoid group. So this is another group that feeds in the larval stages, in the immature stages, feeds on a single arthropod host, eventually resulting in that host death or effective death in the case of these guys. So these are parasitoids of an interesting range of insects that it's hard to find really any common thread amongst these hosts. We have numerous Hymenoptera that serve as hosts of Strepsiptera. The most common of which you'll probably see are paper wasps, genus Polistes, that have these parasites, but also a number of bees also are parasitized by Strepsiptera. And then we also find them attacking the Cicadellidae, and the Thysanura, the silverfish, although this is these groups are pretty rare and you're unlikely to come across them. And then finally some cockroaches are also parasitized by Strepsiptera. The vast majority of species are characterized by having an immobile female. That is, the female is living within the body of its host as an endoparasitoid and basically never moves from that position. The male, in contrast, develops as an immature within the body of the host and pupates there, but then it emerges to fly off and somehow find a female. Obviously, there's probably a good chance that some sort of pheromonal communication is going on for these tiny males. You know, they're usually about 2 or 3 millimeters long. They're not big at all, in order to find the females. Strepsiptera are also characterized by what's called a planidial first instar larva. All a planidium is a well sclerotized, clearly segmented first instar that is mobile. And we find planidia in a wide range of groups. For example, within the parasitic Hymenoptera, we find them in the families of Perilampidae and the Eucharididae, so those ant parasites that I talked about before. And we also find them in the family Meloidae, in the Coleoptera, the blister beetles. So they're kind of strewn around the phylogenetic tree of insects. And so we have this mobile, well-sclerotized first instar larva, but then subsequent molts into the later instars results in the larva becoming less and less motile, usually usually less sclerotized, so a more membranous larva. And this phenomenon is called hypermetamorphosis, where it actually seems to be going backwards in development. That's the kind of a quick brush of what the basic biology is for most Strepsiptera. But actually, the earliest branches, it appears, off the phylogenetic tree of Strepsiptera, we have some forms of biology that are a little bit more interesting and intriguing. In the family Mengenilidae, these are parasitoids of silverfish. Both the males and females pupate away from the host. That is, the mature larva, pre-pupa probably, emerges from the host and pupates in the soil. And the adult females in this group are motile, and I've got a picture of them. Although they are clearly hypertrophied in the sense of being parasites, you can clearly see some more typical attributes of an insect. Recently, I discussed all the controversy over where Strepsiptera belong in the phylogenetic tree earlier, but the recent phylogenomic analysis by Oliver Niehaus et al. Oliver's part of the group of the big one kite group. Uh, he's based in Germany, has published an analysis of the Strepsiptera, phylogenomic analysis that comes pretty unequivocally to the conclusion that Strepsiptera are the sister group of Coleoptera. So Coleoptera and Strepsiptera had a common ancestor from which one diverged and became these highly specialized parasites, relatively depauperate species, and the other became this mass flowering of the beetles. So let's look at a few Strepsipterans. This is a male Strepsipteran viewed from below, and you can see the really characteristic features of these guys. They have what's called posteromotorism, which means that they rely on the hind wings to generate their flight. So these are the hind wings here. I can't see the forewings in this picture. No, that's the forewing right there behind the front leg. I can't see it on this side. That's where I was looking initially. One of the common names for these guys is twisted wing parasites because of this unusual folding in the hind wing of the male. It's very difficult to discern typical venational patterns in these guys. Other typical characteristics are in the males what they call these raspberry-like eyes. So we have compound eyes with large, relatively few and very large ommatidia. This may be correlated with the fact that many male Strepsipterans come out at night, and they can be attracted to light. That's the way I caught my first male Strepsipteran, just sitting out at a campsite in the Everglades with the Coleman lantern on, and in flies a male Strepsipteran. It was pretty exciting. I know I get my thrills pretty cheaply, don't I? And then the final characteristic I want to point out are these what are called flabellate antennae, where basically this is the main antennae here below, and this is just the side branch off one of the basal segments. Other species of male Strepsipterans will have these branches coming off of multiple segments. I'll have an image of a beetle a little bit later on that has just fantastically flabellate antennae. So that's the male. Here is a picture of, so this is a male Polistes, a male paper wasp, one of our local native species, I want to stress. And you can see peeking out underneath the turga of this guy, in the light I can't see more than two of them, I think there's a few more here, are the apices of the Strepsipteran parasites that are living within the gaster or the metasoma of this male Strepsipteran. If this is a male, if one of these is a male, then that male will emerge from the pupa hair and fly off, find a female, and then he'll mate with her. This is the posterior part of the body with the gonopore exposed here. So he will mate with her through her exposed abdomen here, and the eggs will be fertilized there. So that's usually all you see of a female. But remember that family Mengenillidae that I talked about. So this is a female Mengenilla. This is really a very strange Strepsipteran. You can see it has well-developed thoracic legs, which you don't find in the normal endoparasitic forms, like in that Polistes species there. Antennae, compound eyes here. These guys are motile. There's so many details about their biology that remain to be deciphered. It's just a fascinating thing, but these are fairly rare, and obviously that's going to be something very difficult to do field work on. But there's lots yet to be learned about how they evolved into these obligate and very highly reduced endoparasitoids of other insects. So that's really all I wanted to say about Strepsiptera in this context. Let's move on to the real star of this show, the Coleoptera. I'm sure you've seen this quote before. It may be an apocryphal quote, but purportedly J.B.S. Haldane, famous population biologist, geneticist in the early part of the 20th century, was on a show, a radio show, being interviewed, and the interviewer asked Haldane what he thought that the tremendous diversity of beetles told us about the creator. And Haldane is purported to have said that it reveals an inordinate fondness for beetles. Um, Probably, well, definitely elaborated, but I don't know how big a grain of truth there might be in that whole story. But there is one thing that's for sure, is that there are a lot of beetle species. Estimates vary all over the board, but usually they're in the range of somewhere from 300 to 400,000 species of beetles that we know about right now. And as with so many insect groups, we're still discovering new species of beetles all the time. This estimate of 380,000 described species comes from just a few years ago, but there's no census of all the beetles that have been described in the world, and so take this as a roundabout figure, but recognize that this is at least double, if not triple, the number of species in all of the other major, what I call the hyperdiverse or mega diverse orders of insects. And so the other ones, we're talking 100, 120, 130, 150,000 species. Beetles, we've got twice that many that have been described. Now, whether this reflects the fact that there actually are more beetles, or there are just more beetle people studying them, is not entirely clear, and there's a little bit of controversy. But if we take this number, then that means that beetles account for about a quarter of all of the animals that we know about on the planet Earth, which is just a gobsmacking sort of number. The classification, well, the classification is all over the board. Probably it would be safe to say that most coleopterists would recognize somewhere on the order of 186 families within the group. Sorry, I just chased a hemorrhoid off of my computer. It was sitting there. Maybe she'll come back and I can show you. Beetle characteristics. I'm sure you're all familiar with beetles' general characteristics, and the most obvious one, and the one that gives them their name, is the fact that the forewings in beetles are hardened to form these structures called elytra. And the singular of elytra is elytron, so elytra is plural. These are modified forewings. They seem to be primarily associated with providing protection for the hind wings and the abdomen. We'll see that in a second. And they don't provide a significant component to the ability of beetles to fly. I think there have been experiments done that if you actually took the elytra off of a beetle, that it would have difficulty flying. Of course, that's hardly a controlled experiment. There could be many reasons that if you amputate a huge chunk of the beetle, that it won't be able to fly well, not least because you've changed its whole weight distribution on its body. Anyway, they are not the primary source of the forces that enable the beetles to fly. That comes from the hind wings. I've got a picture of hind wings in the variation, which is very bizarre in many ways. But these hind wings, in order to fit beneath the elytra, are folded both longitudinally along the length of the wing and transversely. That is, the hind wing folds back over itself. The usual adaptive speculation, adaptive storytelling for the significance of elytra is that it allows the adults to live in concealed tight spaces, like underneath bark, in the soil, underneath stones, things like that, while still retaining the ability to fly. Whether that's was true or not, who knows? It's certainly true today that you do find many beetles in these cryptic, concealed habitats. So this is the example of a beetle with fantastic flabellate antennae. You can see all these lobes coming off of the antennae here. But I put this picture here to show you the hind wing, the wings, and the abdomen of the beetle. So here we have the forewings that are the hardened elytra of them, and the hind wings expanded out from behind. Now typically, I guess this appears in another slide, but this is a good point to say it, is that this abdominal sterna, the underside of the abdomen of beetles, is very well sclerotized, very hardened, whereas the terga, which normally when the thing is not flying are covered by the elytra, the terga are more weakly sclerotized, flexible, membranous, and so the elytra are providing protection for those terga. Which is chicken and which is egg is unknown, and certainly in beetles that have short elytra, the dorsum of the abdomen is typically much more well sclerotized, much harder to provide for its own protection. Let's take a look at these hind wings. Oh, sorry, one little side point. Those hardened elytra make beetles very conducive to being preserved as compression fossils. This is a fossil beetle wing, and as a matter of fact, this is identifiable to the level of family. And we'll come back to that in a few slides. I said beetle wing. This is a fossilized beetle elytron. You can find these in fossils that go back as far as the Permian era. So beetles are a very old group and very well represented in the fossil record by these hardened elytra. Not always are the elytra able to sufficiently distinctive to enable you to identify a particular beetle to family. This one is, and I'll show you why when we get to a modern representative of this species. So I think now I can safely go on to the hind wings. Right, so here we have a scarab beetle, a rhinoceros beetle, in which they pulled the hind wing off to show the venation here. This oval inset here is this area on the hind wing, and I really wanted to put this up to illustrate a couple of things. One is that, first of all, the obvious thing is that the hind wing venation in beetles is very, very highly modified. We don't see the classical longitudinal wing venation that you think of when you describe insect wing veins. People have worked hard in order to try to homologize what you see in a beetle wing with that venation, and so you can recognize the radius, different branches, radius anterior, radius posterior. Here's the media posterior wing vein. What else do we have here? The cubitus anterior, the anterior anal vein, posterior anal vein, and the jugal vein back here. Notice this little pinch here. This is actually a hinge about which this wing, when at rest, is folded back over itself in order to be tucked under the elytra. So the hind wings can be significantly larger than the forewings of the beetles and still be fitted underneath the elytra when at rest. And so they tuck them back under by means of movement of the apex of the abdomen, the reaching up and pushing on the wing, and also the hind legs, or maybe the middle legs, also reaching back and pushing and pulling on the wings to get them to fold along this branch and then fold longitudinally, well, probably around this point as well. So it's going to fold here and then here in order to be tucked underneath the elytra. So some more characteristics of beetles. They have an enlarged shield-like pronotum, which is really one of the more obvious things when you look at a beetle and you see its thorax from above. What you're primarily seeing is the pronotum. You can get a little glimpse of the meso and metathoracic segments, but they're mostly obscured by the large pronotum and the large elytra, which are abutting them. The pterothoracic segments, so we're talking about the thorax, the segments that bear the wings, the meso and metathoracic segments. These are strongly united in a beetle. In some cases they're fused, but typically they're very, very strongly held together. Whereas the prothorax has a more membranous connection to the mesothorax and is more flexible. I mentioned that the abdominal sterna are strongly sclerotized, while the terga are typically more weakly sclerotized in those species that have elytra that cover the entire abdomen. In families like the Staphylinidae and the many other families that have shortened elytra, then you're going to have the dorsum of the abdomen also well sclerotized. And then finally, very rarely do you find ocelli in beetles. So they obviously have a well-developed compound eyes, but not ocelli. So let's take a little dive here into beetle classification. I don't have the number up here, but there's more than a dozen superfamilies of beetles, and as I said, there's somewhere on the order, experts differ, but somewhere on the order of 180, 190 families. So obviously we're not going to cover all those families or expect you to be able to know all those families. What I expect you to be able to know in terms of beetle families, both primarily in context of what you should be learning with hands-on experience in the lab, is how to find out what beetle family it is, which resources to be able to use, and how to use keys in order to identify them. Classification. Let's step away from families. Let's even step higher than superfamilies in the classification and talk about beetle suborders. Generally, 4 suborders are recognized among extant beetles, so beetles that are still alive. We're not going to get into extinct beetles in this context. These are, well, let me say first before we dive into that, 2 of these are very common and very familiar to you, and the other 2 are very small groups, very rare groups, and in some ways actually quite bizarre creatures. Starting at the top here on this slide, the Adefica is one of the more common, very common, I should emphasize, very common groups. They comprise about 10% of all beetle species. And so if we're talking about 300,000 species of beetles, you're talking about 30,000 species of Adefica. There are a dozen or so families, something on that order, and they are generally predaceous, both in the adult and the immature stages. Classically, 2 groups of Adefica have been recognized. One set of families are all aquatic, and so these go by the name of the Hydrodefica, and the others are all terrestrial, or primarily terrestrial. There are a few exceptions, and these are the Geodefica. We'll dive a little bit more deeply into that in just a few minutes. One of the very small, in many ways bizarre groups is the Archostemata. I think there's only 2 families of Archostemata, and only about 35 or so species that are still alive today. They're very well represented in the fossil record, but only about 35 species there today. So these include the reticulated beetles, and I don't think that the Micromalthidae has a common name. Then there's another very small group, one that most people, I don't know that I've actually ever encountered any representatives of the Myxophaga. There's a couple of very small families that occur, a grand total of only 65 thereabouts species of these things that are aquatic or semi-aquatic beetles. So not all aquatic beetles are in the Hydrodefica. There are aquatic beetles in the Myxophaga, and also in the next group, which comprises the vast majority of beetle species, the Polyphaga. So we've got 4 suborders of beetles. People are pretty well set on recognizing those as monophyletic groups. But the interrelationships among them are still a bit uncertain. We've made a lot of progress. If you just look over the literature, you've got 4 suborders, but there's been 9 different hypotheses proposed about how those 4 groups are interrelated. That what I think the maximum would be 10. So we pretty much got evidence that might support each and every one of those. But if you look at the recent, more comprehensive analyses of beetle phylogeny, ones that rely not on morphological characters but on molecular characters, they all seem to to come to a common conclusion, even if the support values are not exactly as high as we might like to see them. So I assigned McKenna et al. 2015 for you to read. It's actually been superseded now. I'm probably going to have to substitute out. I might want to substitute out some of these other papers for future classes, although these tend to be a little bit more specified. What's the word I'm looking for? Specialized in what they're actually asking the questions. But Dwayne McKenna's et al. Big list of co-authors looked at 8 genes across 367 species of beetles that represented about 94% of families. So in terms of family representation, they had a lot of families but relatively few genes. No E on the end of dong, sorry about that. They increased the number of genes, kept the number of species they had in their analysis about the same, but we see a 30% or so drop in the number of families that were represented in that analysis. And then just a couple of years ago, Dwayne came out with another paper, again with a whole long list of collaborators, and this represented one of those sub-projects off of the one kite phylogenomic analyses. So this is based upon both transcriptomics and whole genome sequencing of beetles. They identified over 4,800 orthologous genes within the 146 species of beetles they had in their sample. But see, now we're down to only about 50% of the family. So this high representation is kind of why I still have assigned this paper for you to read. But this is the one that has the most comprehensive number in terms of genes that have been analyzed. As I said, they all come to the same conclusion, although without the absolute statistical certainty that some authors are looking for, and that is that we have, first of all, the beetles branch into two groups. On the one hand, we have the Polyphaga. That's 90% of all the species of beetles in the world. And then we have the other group that's made up of the other 3 suborders. Within those 3, the 2 that are most closely related are these 2 bizarre small groups, the Archostomata and the Myxophaga. And then they are sister to the Adephaga. So this group comes together first, then the Adephaga, and then they join with the Polyphaga. Here's the phylogenetic tree for McKenna et al.'s recent phylogenomic analysis. Let's home in on the basal part of this tree to see its structure. Well, just taking an overall look right now, in terms of where the Polyphaga are here, the Polyphaga go from here all the way up to this point on the tree. So all of these are in a single suborder, and the other 3 suborders are in this area of the tree right here. So let's focus in on this general area to look at the branching pattern. Okay, so here we are at the base of the tree. The outgroups that they used in their analysis were Strepsiptera and Neuropterida. If you can read over here, there's a Rhiphidioptera in there, Inocelia. I'm not familiar with the name Subilla. Corydalis, so that's a megalopteran. And then Osmilus and everything down to this point right here with Triosocera. Just checking out to Euroleon right here. These are classic Neuroptera. Then we have the Strepsiptera, represented by an interesting from Mengenilla. That's that weird thing that I showed you a picture of the motile female. And then some more classic Strepsipterans. And they form a monophyletic group. Absolutely no surprise there, given their bizarre morphology and life history. But they are sister group to the rest of the Coleoptera. I should point out here that down at the bottom of that image, they had a time scale. So what these gray bars are showing is the degree of uncertainty in the dating of these branches on that time scale. So let's see, where was I? We got a monophyletic Strepsiptera that is sister to the rest of Coleoptera. That paper by Nihus that I mentioned, demonstrating that Strepsipterans were sister to Coleoptera, came out a few years earlier than this publication. Okay, so Streps sister to beetles. Whoops, there we go. Then within the beetles per se, sensu strictu, we have a monophyletic group here. Here's the Adefica forming here, starting with, oh no, I'm sorry. This is the combination of Adefica, Archostomata, and Myxophaga. So starting with Priacma and going down to Carabus. And then down here at the other half of this branch are the Polyphaga, which I've colored here in green. And so if you remember on that image of the page, from here on down, that's all Polyphaga. That was the majority of the representation in their tree here. And if we look up, let's see, I didn't put arrows on this. So we have this, which is Adefica plus Myxophaga plus Archostomata. This is Adefica down here. And here is Archostomata in this branch and Myxophaga in this branch here. So some of these, I told you some of these Archostomatan groups are really weird. This is the all but one species of living Archostomata belong to this family, the Cupetidae, or the reticulated beetles. These are pretty unusual, rare beetles, although it's been discovered that for some reason, some of these guys are attracted to a compound that is in bleach that you use for washing. And so one of the ways they discovered that you could collect them would be by washing your sheets in bleach and then hanging them out to dry. And for some reason, the Cupetids are attracted to these things. It's a little bit bright where I am, so it's not clear. I hope it's clear to you. You can see this interesting pattern on the elytra here. This is why they're called reticulated beetles. Reticulated means net-like. And so you see this distinct pattern on them, which is what I showed you earlier in this fossil. You can see this same rectangular pattern of rectangular cells arranged in rows on the elytron. So this is a fossil Cupetid. Cupetids are very common in the older fossil record, perhaps overrepresented in part because this pattern of reticulation on the elytra is so distinctive. Not much known about their biology, except that they are wood borers in the larval stages. The other represented family in the Archostomata is the Micromalthidae. The Micromalthidae has but a single species that's ever been discovered. And so I'm talking about, there are numerous fossils of Micromalthidae, particularly in amber. All of them seem to be the same species as the species that's still alive today. This is Micromalthus debilis. You can see the size of this creature is very small. It's only about 2 millimeters long, and it looks kind of, unexciting, nondescript, until you start looking at their biology. Let's see if I can explain this. Micromalthids, as larvae, bore into wood, and one of the common names of them is telephone pole borers, because back in the day, probably before telephone poles had such heavy impregnation with creosote and other preservatives, you could find these tiny beetles boring in telephone poles, but they also bore in hardwoods, typically wood that's starting to decay, and you find these things. But that's not bizarre. There's lots of beetles that bore in wood. We're going to see a bunch of them here. But what's interesting is that for most of their life history, these beetles persist in what appears to be the immature stage. So they persist. Here we have a larva, and most of their life history goes through this part of the cycle, the overall cycle right here, in that the larvae produce offspring themselves in the forms of more larvae, which then molt. They go through a little bit of metamorphosis here, going from what's called a cerambycoid larva. So a cerambycoid larva obviously is something that looks like a cerambicid larva, the cerambicity being the round-headed borers, a very common family. Well-developed head, no thoracic legs, things like that. So basically an apodous, legless larva, which molts, becomes this pedogenetic larva. Pedogenesis is the reproduction by the immature stages. And so they go through this cycle here. But once in a while, driven by environmental cues, perhaps, one suggestion is that when the wood that they're in starts to dry out, these things can produce an egg, which then produces a curculianoid larva. So this looks like the family Curculionidae, or the weevils, with small thoracic legs, which interestingly, well, I suppose I should have gone up here to the female first. Sometimes this is not the cycle we go through, but this larva will molt into a pupal stage and produce a female. Rather normal, though, although there's no egg that goes along here. The egg here produces this curculianoid larva, which when it hatches, this larva turns around and starts feeding on the abdomen of its mother. It starts consuming the pedogenetic, this larva here, that produced this egg. And after about a week of eating its own mother, it then pupates and becomes a male. One of the really bizarre things is that you see these dotted lines here. No one's observed whether or not males and females actually mate and produce eggs. I even saw one reference that said that they are sterile, that they cannot produce eggs. And so all of the reproduction is in this pedogenetic stage here. So one species of micromalthids worldwide. If you look at all the fossil records, they're all the same species. And I did let that word slip out, worldwide. These things are found all over the world. One of the earliest branches of the phylogenetic tree of beetles. In terms of its adult morphology, it retains lots of characters that are plesiomorphic for beetles as a whole. But obviously, this life history is just way too bizarre. It's just gone off on its own way. And you know, it's survived for tens of millions of years. It must be even longer than that. If they have cupetids back in the Permian, then we're talking about this creature having survived hundreds of millions of years with this very bizarre life cycle. It's not common, to be sure. It's a pretty rare creature, but it's obviously successful enough to have persisted all the way along. So let's go from, well, we won't go from bizarrity to bizarrity, but let me introduce a representative of the Myxophaga. This is the family Hydroscaphidae, and I don't know if you can tell, but this is a small aquatic beetle. Some are associated with very rapidly running waters. They're feeding upon algae, mosses, and things in the general area. Again, very few species in this group, and very restricted in their distribution. So let's get into the real meat of the beetles, where all the diversity really lies, and start out with the Adephika. Remember, I told you that the Adephika comprise something on the order of 10% of all beetles. So we're talking 30 to 40,000 different species of beetles here. And many of these are going to be extremely familiar to you. As I said, the classic classification divided them into two groups. The Geodephica, which is, well, it depends upon your classification, is almost entirely composed of the family Carabidae, or the ground beetles. Although there's been a little bit of a bouncing ball going on here in terms of the tiger beetles. When I first took taxonomy, the tiger beetles were, excuse me, were recognized to be closely related to ground beetles. And then people uh, came to the conclusion that the tiger beetles represented a grouping within the ground beetles. And so as to not make the Carabidae paraphyletic, they subsumed the cicindelids within the Carabids as a subfamily there. Well, I just saw a paper this year that has extracted them yet again as a separate family. We'll keep our powder dry as to whether or not we'll uh, buy in to that conclusion. But you'll see cicindelids referred to both as, well, as cicindelids as a family and also as cicindelines as a subfamily. In the Hydrodiphica, we have lots of very familiar groups of aquatic beetles, the Dyticids, which are the predaceous diving beetles, Gyrinids or whirligig beetles, perhaps less familiar to you because they're much smaller in body size, are things like the heliplids, the crawling water beetles, the notarids, the burrowing water beetles. According to that latest phylogenomic analysis by the one kite subgroup, McKenna et al. 2019, the Hydrodiphica do not form a monophyletic group, whereas the Geodiphica do. So just to show you a few images of some more or less classic sorts of beetles that fall into this grouping. The Carabidae are the most diverse and a very, very well studied group, very popular. This is one of the classic Carabids. This is the genus Calosoma, and that name literally means beautiful body, and you can see why they would call it that. These guys are quite large. I would guess maybe 5 centimeters in size. They get to be very large. Predaceous as adults, both adults and larvae, and sometimes species in this genus have been, people have attempted to use them as biological control agents of things like gypsy moth caterpillars. I believe they introduced a species of Calosoma from Europe in order to try to control the gypsy moth. Didn't really work. Remember back to our very first, I believe it was our very first video when I introduced you to Terry Irwin. This is a species of Agra, the group that Terry studied. Although the common name for the family Carabidae is ground beetles, in fact, beetles like this genus, and there's lots of species, are all arboreal. That is, they're living up in the trees. So we find Carabids in a wide variety of habitats, and we need not take their common name too literally. There's species in the trees, there's species on the ground. There's also some species that are semi-aquatic or associated with waters. So there's a tremendous amount of variability. And because these things are large, active, and in many cases quite beautiful, there's a lot of interest in ground beetles, both professionally and at the amateur level. So we know a lot about ground beetles. A couple of representatives of the family, now the family Cicindelidae of the tiger beetles. So as I say, you'll see these classified both as ground beetles and in their own family. Here we have one that has, this is a rather somber colored tiger beetle with the orange spots on the back. Some of them, like the next one, are positively incandescent almost with the metallic colors that they show. Maybe you can get a sense of it from the stance that this creature is adopting here, or maybe you've tried to catch tiger beetles. These are extremely rapid predators, both running and flying away, and they're very difficult to catch with just a net. I mean, it can be done, but they are very, very fast runners and also flyers. So very, very good flyers. If you're familiar with the species around here, you mostly see individuals, and they're very very typical of areas with sandy soils, because that's where the larvae live. The larvae burrow down into the soil with just their head at the top of the burrow and reach up and snag insects, and probably other arthropods that are passing by. Both of these are voracious predators. These things could give you a very nice nip if you were foolish enough to stick your fingertip in between its mandibles. I wanted to mention one other thing about these. Our local species are more or less solitary, but you can find these things that they form actual swarms. I was collecting along a stream in India, and there was all these insects flying and in a big blur, and they were rather large. By the time we were able to catch one, we discovered to our surprise that they were tiger beetles that were swarming there. And so I think we come to generalizations, conclusions about what our locally common species do in terms of their biology, and recognize there's a lot more interesting things that are going on out there in groups in other parts of the world. To the Hydrodephica, some of the water beetles, we have the predaceous diving beetles. As the name implies, both the adults and the larvae are aquatic. The larvae look in many ways similar to the megalopteran larvae that I showed you. I don't have pictures of the larvae, just the adults here. But they're, I mean, a key to aquatic insects will quickly get you straightened out as to whether it's a megalopteran or a coleopteran. Predaceous diving beetles, one of the ways, there's another family that you might confuse these with, the Hydrophilidae that occurs in the Polyphyga. But one of the ways that you can easily distinguish Dyticids from Hydrophilids is the manner in which the adults swim. You see the hind legs here on this side, and here it is over here on this side, have these extremely long hairs protruding out of them. And so these are basically their paddles, which they're oars that they are stroking to move through the water. And a Dyticid will move the hind legs in synchrony, swimming those together, whereas a Hydrophilid has the legs going alternately. So a very easy way to quickly and at a distance distinguish them. A big guy like this, you're not going to have any problem with. But some of the smaller species, a Hydrophilid at first glance can look very similar to a Dyticid. Another one of the very interesting things about these beetles is that in the males, in a number of different groups, the forelegs, the four tarsi actually, are expanded. So here we see the apical two segments of the four tarsus, and this is the base of the tarsus. It's expanded into this wide, almost circular pad, which is used to assist the male in grasping and holding on to the slick elytra and pronotum of the female as he attempts copulation. So this is the foreleg, mid leg, and this is the hind leg, which is providing all the power for swimming. And you can see how the femora are greatly enlarged here, and you can't see it because it's not in focus in this image. The coxa is also greatly enlarged. So there are massive muscles that are originating in the body and in the femora, and then inserting here in order to move these legs to provide them propulsion through the water. They're very good swimmers. You'll have to take the aquatic entomology course. You would get into much more detail about how predaceous diving beetles and other Hydrodephica actually are able to get oxygen while they're underwater. I'm not going to get into that here. There are too many good beetle stories to try to indulge myself in all of those. Already, I've probably indulged myself too much. The family Gyrinidae are the whirligig beetles. Here we have one. These guys, they can dive underwater, and the larvae live underwater, but the adults are most commonly seen on the surface of the water, where they're paddling about. These are the forelegs. They're not using those so much for paddles. I think hind legs are visible in the next image. Yeah, so here we have a whirligig beetle. Here's the forelegs. And you can see the mid legs and the hind legs are actually short, but they're very broad. It will be an interesting exercise for you to homologize the parts of the leg. Here's the hind coxa. And one of the characteristics of Oediphaga is that the hind coxa overlaps the first abdominal sternum. So here's the posterior margin of the first abdominal sternum. The anterior margin is up here somewhere underneath this leg. I can't see it. But the hind coxa overlaps it. Trochanter, femur, must be tibia, and this must be all the tarsal segments, so into these wide flattened paddles. And so they're swimming a combination of the mid and hind legs rapidly. If you ever tried to catch a gyrinid individually, they're very, very difficult to catch. They're excellent swimmers on the surface of the water. One of the other features that's characteristic is that here we have a beetle seen from the side, foreleg here. They have antennae, but their antennae are very small. But anyway, the point I want to make is that they have the compound eye subdivided into two physically separated units. One part that lies outside of the water, so it's seeing through the air, while another part of the eye is underwater and seeing through the water. Must have something to do with avoiding that layer, the surface layer itself, and the refraction associated with that makes us think that something that's sticking into the water is actually bent because of refraction in the water. That would be my guess anyway. So let's dive into, or let's take and dip our toe into the Polyphica. As I said, this comprises more than 90% of all the beetle species in the world. This tremendous number of species is matched by tremendous diversity in their biology. In terms of trophic levels, we have predators, we have pollinators. For example, the giant water lilies in South America are actually pollinated by scarab beetles, which are polyphagans. Lots of herbivores, detritivores, fungivores. If there's something, didn't include scavengers here as well, which is also another very interesting niche that they've moved into. In the McKenna et al. Analysis, they recognized 18 superfamilies of Polyphica, 2 of which are especially diverse in the terms of number of species. These are the Chrysomeloidea, so the leaf beetles, the round-headed borers are the major groups here, the leaf beetles being the Chrysomelidae and the round-headed borers being the Cerambycidae. 2 very closely related families that can, in some cases, be difficult to distinguish. Those, together with the Curculionoidea, which includes the weevils and all the related groups around weevils, make up a monophyletic group called the Phytophaga. So within the Phytophaga, these are all feed upon plant materials. This plant materials may be leaves, they may feed externally on leaves, they may be leaf miners, just like the elm leaf miner in the Hymenoptera I showed you before, burrowing between the upper and lower epidermis of a leaf and eating all the tissue in between. Wood borers, things like that. Over 125,000 described species, and there's certainly many, many, many more that are yet to be described. In many circles, the Curculionidae, the weevils, are thought to be the largest family of insects alive today. In Duane's recent phylogenomic analysis, one of the things I said, some of these papers were a little bit more specialized, and this is one thing I wanted to refer to, is that they were particularly interested in looking at the beetle phylogeny in order to look at a couple of families of enzymes that are associated with herbivory. And these were what they call G32 invertases. So these are enzymes that will take sucrose, which is the primary product of photosynthesis in angiosperms, and break sucrose, which is a disaccharide, down into its 2 components, glucose and fructose. And there's a whole other family of enzymes that they call the PCWDE, an acronym that simply means plant cell wall degrading enzymes. They found that these two families of enzymes were elaborated. That is, there were multiple copies, multiple paralogous copies of these enzymes in two major groups within the Polyphaga. One of those is the Phytophaga, so the chrysomelids and the weevil families, and the others in the superfamily Buprestidae, which you may recognize that name for the family Buprestidae, which are the flat-headed borers. So familiar things such as the emerald ash borer is a Buprestid. So in these two families, well, let's do it this way. In the other families, most of them have a couple of different GH32 invertases, but don't have the plant cell wall degrading enzymes, or maybe sporadically there might be one enzyme appearing in a family. Whereas in these two groups, they had numerous different genes for both of these families of enzymes occurring there. And when they did their analysis, a gene tree analysis, looking at how those genes were interrelated, and including not only beetles but other organisms, they came to the conclusion that these two beetle groups independently gained these enzymes by a means of horizontal gene transfer from bacteria or fungi, so that they were somehow in the course of living in close association with bacteria or fungi that were capable of, for example, breaking down lignin or breaking down cellulose in a decaying tree. Somehow those genes became transferred into the genome of these species. So by virtue of being able to look at whole genome sequences, they could find these enzymes, the DNA that codes for these enzymes in the genome. I mean, if you're doing the transcriptome, there's always the possibility that what you're seeing is a contaminant of, say, a bacterium that's living in the gut. No, these things are actually encoded in the genome. So how they got there, why they got there is a mystery, but they appear to have come from bacteria and fungi. And both of these groups are characterized by high levels of diversification. I've already talked about how many species of Phytophaga there are. There are also lots of species in the Buprestidae. And so their suggestion is that this correlation might imply a mechanism that by enabling the ancestral beetles that picked up these enzymes into their genome and therefore could live independently of the bacteria and the fungi, opened up a new niche for them, a new hitherto unexploited niche that they were able to move into and then have adaptive radiation take over, and resulted in the large numbers of species. An intriguing hypothesis that opens itself up to lots of interesting ways to test that hypothesis, which of course is the value of these kinds of phylogenetic analyses. Okay, let's take a quick run through some of my favorite beetle stories. The first is in the superfamily Staphylinoidea. There are many species of the family Staphylinidae, but another family in that superfamily are the Sylphidae, or the carrion beetles. And I wanted to mention them because we normally associate social behavior in insects with, well, the termites, of course, and the Hymenoptera. We talked a little bit how there are eusocial thrips and aphids, individual groups here and there. But the sylphids, the carrion beetles, also exhibit a form of social behavior. Carrion beetles, as their name implies, use carrion as the food source for their larvae. And so what we're seeing here in this probably very smelly picture, thank God there's no smell-o-vision here, we see two adult beetles, most likely the male and female of this brood of larvae here. And what the adults orient to a small carcass they may have found, let's say a mouse or a turkey chick that has died or something like that. And upon finding that carcass, they dig beneath the soil below it and gradually submerge the carcass into the soil, cover it up, and then masticate, and probably are releasing digestive enzymes here as well. Things probably loaded with bacteria as well, to convert the former carcass, looks like from the hair around here, this was a mouse or a vole or something on that scale, into a ball, make a little cup-like indentation in one end, lay their eggs from which the larvae hatch. And the adults will actually feed the larvae. I think I have that in the next slide. Will actually feed the larvae some of this material that is decomposing. So we see cooperative brood care, well, in this case between a male and a female. We don't observe the other hallmarks of eusocial behavior, the reproductive division of labor, the overlap of generations. This is a single generation because this is an ephemeral food source. This is not going to last more than a few weeks, likely. So this is called subsocial behavior, when you have the adults actually feeding and rearing the young within this ball. What do I have next? Within the Polyphaga, in the superfamily Elateroidea, we find numerous species in which there is bioluminescence. That is, the organisms themselves can produce light. The most famous example of this is, of course, the family Lampyridae. And here we see most likely, well, it could be a male or a female. I'm not that familiar with Lampyrid taxonomy. With the apical abdominal segments, looks like in this case the 3 segments with internal light-producing organs. And of course, I'm sure you are all familiar with the story about what these light-producing organs are used for. But I want to emphasize, first of all, that these light-producing organs are not limited only to the adults. So when I said that, you probably thought, oh yeah, males are using these to signal to females, the females signal back, and then they find each other through these photic signals in order to mate. This is the light-producing organ. I believe this is a female. You can see the female is in this species is very, let's call it larviform. Looks like a larva. I almost confused myself there because the larvae also are not photosynthetic. What do I want to say? Bioluminescent. There. So obviously the larvae are not using them as mating signals. This is a time-lapse photograph of what you're seeing is a beautiful image of male fireflies flying through the air, signaling. The females are either on the vegetation or on the ground, signaling back to them. And perhaps you can see some of the flight pattern. It looks like we have a male here that's flying and going up and down in sort of a sinuous pattern. So the signals can be differentiated on the basis of the wavelengths that are produced, the flight pattern of the male, doing the sinuous pattern, or sometimes they'll get my hand in the image here. They'll just emit light as they're flying up and up. So you can distinguish different species that way. And you get these beautiful patterns here. I encourage you, when the fireflies emerge, oh gosh, probably 4 or 5 weeks, we'll start to see fireflies. Try to, when you see an individual flying, try to trace its path through the air. In those dusky hours where you can still see the individual beetle flying, and detect when it's releasing its signal of light. I had a little bit of trouble in my initial recording of this video to get the next slide, which has a video on it, to get it to run properly. So this has been inserted after the fact, and hopefully it'll run correctly. What you're going to see is an example of what's called synchronous flashing. on the part of fireflies, whereas the fireflies we have around here, everybody's flashing more or less independently. In parts of the world, and most famously in Southeast Asia, the fireflies are watching each other and will synchronize their flashes so they're all going on and off at the same time. And so I found a video illustrating this, which stimulates this on the part of the fireflies by using LEDs. So you can kind of get the fireflies to meet your own timing by flashing the LEDs at a particular frequency. Anyway, I thought I've always wanted to see this. If my former student in Thailand ever invites me to go to Bangkok, then I want to figure out some way to get on a boat and go upriver and see the synchronous fireflies in their great show. So that's what's coming up in the next segment here. So here we have another member of that same group, the Elateroidea. In fact, this is a member of the family Elateridae, the click beetles, to demonstrate that it's not only the fireflies, the lampyrids, that are bioluminescent, but also you see this animal has a pair of light organs on either side, in this case not on the abdomen, but on its prothorax. This is actually a larva, and the common name of this larva is the railroad beetle. Some of them have red bioluminescent organs on their head, on the front of the prothorax, which gives them the appearance of a train here. This is a larva. Here's the adult. That's the family Phengodidae. Here's another absolutely gorgeous flabellate antenna with those wild extensions coming off of the basic segments here. Huge eyes, the better to see you with, my dear, at night. Short elytra, and so these things are going to have a more sclerotized abdomen. All of these are bioluminescent, so this occurs widely throughout the coleoptera. And then finally, let's take a brief look at some of the curculionids. So this is a weevil. You recognize it from the elongate, what's called a rostrum, an extension of the head. The the mouth parts are not back here on this part of the head. They're at the very tip of this rostrum. And so it uses this. Typically what they do is they chew a hole in whatever substrate they normally, the larvae normally live in, put their eggs in there. And so this is a means, whereas a hymenopteron might have an elongate ovipositor in order to reach into a plant in order to put its eggs, the weevils are using their head elongated in order to place their eggs. I believe that this is a species of the genus Curculio. And you may know that these things will lay their eggs in acorns and nuts and things like that. And we had some species of Curculio that would oviposit in American chestnut. There are two species, one of which was relatively more prolificous. That is, it will breed in other species of plants. But there was one species that appeared to be restricted to feeding on the American chestnut. If you know what a chestnut looks like, the nut you buy in the store, as it's growing on the tree, is covered by an outer husk that has lots of spines coming off of it. And so they would use this long rostrum to reach between those spines, chew out a hole in which to lay their eggs, and then the larvae are going to live within there. Well, the species, as you know, the American chestnut is, I guess one way I've seen it expressed is that they're reproductively extinct at the moment, that there are very, very, or nearly so, there are very, very few American chestnut trees left. There's lots of rootstock left from the days when the chestnut blight was introduced, and so we have lots of volunteers and plants and stems growing from the rootstock, but typically these never reach maturity to produce chestnuts, and so we have no chestnuts. We also seem to have lost through this extinction the insects that were specialized on chestnuts. There are a couple of species of moths, and also apparently the chestnut curculio, which had just an absolutely impressive rostrum that it could reach between those long spines on there. Except for a couple of specimens that were collected in Maryland in the 1980s off of chestnuts derived from a tree that has since died. There have been no chestnut curculio collected and deposited in insect collections in the US and Canada since, I believe the date is 1952. And so we've probably lost this species, even though we had a subsequent introduction of Asian species of the same genus, Castanea, that wasn't attractive to the chestnut weevil, and we've lost it. Now, when I was an undergraduate, the beetles that I'm going to close off with here were all classified in their own, actually two families. The bark beetles were put in the Scolididae, and the ambrosia beetles were placed in the Platypodidae. These are now considered to be subfamilies of the Curculionidae, the weevils. And although I claim to be a hymenopterist today, I actually began my career working as an undergraduate on this creature, a bark beetle, Scolitis multistriatus. Its common name is the Lesser European elm bark beetle. If I remember what I was taught back in those days, this is a male. How can I tell? Well, I don't look at that end. I look at this end, and this golden fringe of hairs on the fronds is characteristic of a male. A female has far fewer hairs and doesn't have that beautiful golden color there. So, I'm not sure how familiar you are with bark beetle biology, but this is a tree from which the bark has been removed. So that's the first thing to note, I guess, is that to call these bark beetles as a common name is a bit of a misnomer. They do live beneath the bark, but they don't feed on the bark itself. Rather, what happens, excuse me, imagine that this tree is covered with bark, and the typical tissues, so what we're seeing here, the wood, is secondary xylem, all right? And on top of that, you would have primary xylem, vascular cambium, primary phloem, and then outside of that, the secondary bark and cork, secondary phloem and cork that forms the bark, the true bark of the tree. In the case of the elm bark beetle, the female finds an elm tree. They're pretty specific to elms, and they particularly love American elms, which is part of the problem. The female bores through the bark down to that cambial layer, so that interface between the phloem on one side, the xylem on the other side, and the vascular cambium in between, burrows straight in. And while in there, she releases a pheromone that attracts males. They mate, and once the female has been inseminated, she then digs a burrow going vertically, and along the way, she'll cut a little niche in the sides of the burrow and lay an egg on either side. And this radiating pattern that you see coming out from the side here, all the way, oh, it comes all the way out here, doesn't it? Are the burrows of the larvae. Oh, here they are right here on that side. I hadn't noticed that before. The larvae that burrow out at right angles to this burrow that the female has made. So this is a very characteristic pattern of the lesser European elm bark beetle. Vertical burrow, and then coming out at right angles to that are the larval burrows. There's a native species of elm bark beetle the gallery that the female excavates is horizontal, and so you can tell the two apart. So, as its name implies, this species was introduced into the U.S. from Europe, and the beetle itself is not that dangerous, in that this is a very small amount of feeding damage that takes place. But one of the things that the beetle does is that within the pronotum, it has a couple of cavities within which a fungus is stored, and the beetle inoculates this fungus into the tree. The fungus gets into the phloem, and is then carried throughout the rest of the tree, effectively blocking the transport of what does phloem carry? The photosynthates in the tree, and resulting in its death. And here we see some of the first indication. This is called flagging, where we have the green leaves of the healthy part of a tree, but somewhere back upstream of these leaves that are dying, the phloem has been blocked such that these leaves are not receiving the transport has been blocked, and this part of the tree is dying here. So that's the first indication, and this can quickly kill an entire tree. And this was a particular problem. The reason that my advisor got involved with studying elm bark beetle back in the day was that it was very typical for urban foresters, so the people in charge of planting trees along streets, to use elms. Here we see an elm that's not doing very well at all, but they have this characteristic, they call it a vase shape pattern that the tree grows, and they were very commonly used as street trees. Kind of get an idea of the age of this photo from that car. Here's another picture a little bit later, and you can see how they would grow on either side and shade the streets underneath. So they were very popular. And so the introduction of this smaller European elm bark beetle and the fungus that it carries along with it has absolutely devastated the American elms, such that there are a few places where you might be able to find a few hangers on, but the vast majority of American elms have been extirpated. People have tried to replace them with other native species of elms, which generally do not grow to this large size, or with Asiatic species, the Chinese elm, for example, or Siberian elm. They also do not grow to this majestic size that the American elm used to grow. This is a gallery, that's what these burrows are called, of an ambrosia beetle. And I can tell this is an ambrosia beetle in part because this dark staining is actually the result of a fungus that the ambrosia beetle has introduced. And they're called ambrosia beetles. Well, the second thing is that the larval galleries stop and are very short. They're called ambrosia beetles because they feed upon the fruiting bodies, the ascospores, I believe, in most cases, although there's a variety of fungi that are used. They'll feed upon those spores, and actually the adults will feed their larvae on those spores. So just like the carrion beetles, there are degrees of sociality that have been evolved within the ambrosia beetles as well. And they come in just a fantastic array of shapes and sizes, and you can see how maybe you caught a glimpse of that in that elm bark beetle, but how the posterior part of the elytra is often excavate. This one is kind of truncate. This one has spines. This one's got a pair of spines. This one's got like a scooper on the end, with which it's probably mostly used to push the frass out of the gallery as the female, or in this case, the male and female, excavate it. So frass is a combination of feces and the wood that they have chewed off to push it out of the gallery so it's freed up. So here we have a bunch of larval ambrosia beetles, more over here, that are fed by the adults. So again, subsocial, and there are even more elaborate forms of sociality occurring in ambrosia beetles. Very fascinating group. So I've warbled on long enough about beetles. They're, as they used to say on the old TV show, there are 1,000,001 stories in the naked city. Well, there are at least 380,000 stories in the order Coleoptera, which we've only scratched the surface of them. What do I want to be the takeaway lesson from this video? So we start with the Strepsiptera, parasitoids with winged males and generally immobile females, although there are some very interesting and rare exceptions. And with the planidial larvae, so this motile larva that somehow finds its host, burrows in, and then undergoes this hypometamorphosis to become a sessile larva, either a male or a female. Within the Coleoptera, and they're the sister group to the Coleoptera. Within the Coleoptera, we have the 4 suborders that are generally recognized, the Archostomata and Myxophaga, the 2 very rare ones, and then the Adefica and the Polyphica, which make up the vast majority, practically all, of beetles. The Adefica are predaceous both as adults and larvae, and includes familiar things like ground beetles and tiger beetles, the families Carabidae and Cicindelidae, along with an array of aquatic beetles, Dytiscidae, Gyrenidae, Haliplidae, Noteridae, and the like. The Archostomata is a very ancient group going back to the Permian. I forgot to mention that in McKenna's phylogeny, their molecular data suggest that beetles actually arose in the Upper Carboniferous, which kind of makes sense. If we have beetle fossils in the Permian, then the very first beetles came earlier, and perhaps as far back as the Upper Carboniferous. Some of these Archostomata, especially Micromalthus, just have bizarre biologies that we really don't understand if they ever actually, males and females. Well, why do they have them if they're sterile and don't even reproduce? It's a bizarre sort of system. Within the Polyphica, that's 90% of the beetles, and the one major grouping, the Phytophaga, includes over 125,000 species itself. And the interesting thing that came out of the McKenna et al. Phylogenomic analysis is that the diversity in Phytophaga, and then also the buprestoids as well, is correlated with an elaboration in the 2 gene families for those invertases to be able to digest sucrose, and the plant cell wall degrading enzymes to be able to break down the lignin and the cellulose in cells walls. That's correlated with the great diversity in those species, and it's something that they appear to have gained. They appear to have gained the genes for these enzymes from horizontal transfer from bacteria and fungi that are living in those same environments. The mechanism, of course, is entirely unknown. So apologize for the little glitch on the Lampirid synchronous flashing. I'm going to put this video into a video editor and insert that sequence in there. So if I do it well, then my comments here are probably totally opaque to you and don't even know what happened, but I had trouble getting it started. So with that, I'm going to call it a day and get to editing the video. We'll see you next time, and until we do, take care and stay healthy.